I think it's really important to bring the AOPs, leave it not only with the regulators, but bring it into the academic world, because we are the ones who can really fill gaps. I think this is one of the major issues that I've learned from AOPs. Where are the gaps and how can academy fill it? I'm not really going to talk about how is an academic forming an AOP, because what I learned through my work with the European Food Safety Agency mainly is that building AOPs is really so much work that it goes beyond the time slots that an academic researcher really has. I mean, we're trying to do this, and I show you how. So how do I implement the AOP work into my daily kind of business? But really getting an AOP ready for submission to the wiki is such an incredible amount of work that it's really unreal. I mean, we built actually 2 AOPs within the last EFSA working group on plant protection products and the development of Parkinson's disease and childhood leukemia, and it took like 10 people to work for a year, and the document that we ended up with, which is published now, is several hundred pages, and it really takes every key step of the biological plausibility and so on into consideration, and it really takes a full-time job to work on this. So this is, I think, the downside of the AOP. The biggest downside is really, besides the linearity, that it's so time-consuming that it's very, very hard to really get into the business on an OECD level. But nevertheless, I think academics also have the abilities to work with the AOPs, and this is what actually I'm trying to bring you a little closer this morning. So what do I do with AOPs? For one thing, I really try to teach what is an AOP, and I mean, not go into the details, that's too much for students, but actually it's important that students have heard about the AOP concept, and it's not, at least in Germany right now, it's not mandatory, or it's, you know, when I say, well, people go to a class teaching, have you heard about AOPs? They usually say, no, not really, you know, and they study toxicology. So I think it's really important that the education also implements the AOPs. And the second one is the academic research. Well, yeah, I try to fill gaps, or when I find new pathways, I'm trying to make my students think about it in the AOP context already, which is not that much different from how we have been publishing papers to begin with. I mean, a very well scientific paper on signaling that we used to publish, I don't know, 10, 20 years ago, contained everything that we actually grab in the AOP in a more systematic way, and, you know, organize it into the scheme that a regulator can use it. I think that's the major thing, that the academics understand that if they want their work to be used, they have to put it into a framework that a regulator understands. So, and this is what I'm trying actually to bring over to the students. So for the teaching, I bring you an example which I always find it's kind of entertaining, and the students understand, I think, easily what the AOP concept is about. And what I use is actually a paper from Nature from 1988, and it's coming from my university, so from the University of Düsseldorf, and the title is "A New Parameter for Sex Education. There is concern in West Germany over the falling birth rate. The accompanying graph might suggest a solution that every child knows makes sense." And this paper has really received a lot of attention because epidemiologists have used it up and down, and it's been discussed in the web for many, many years, because it's showing the difference of correlation and causality. So when you look at these 2 graphs here, so the black dots, it's actually it's the number of newborn babies in West Germany between 1965 and 1980. And the open circles are the pairs of breeding storks in the same area. So looking at the graphs, you could say, oh, okay, so the falling birth rate may be caused by the falling number of stork pairs because the storks bring the babies, right? So, and actually, when you do some numbering with the axis, so it's about 600 babies per year per stork pair, and that's kind of reasonable, right? I mean, in 356 days, the storks should be able to do this. And so our hypothetical AOP would be, if the storks are really bringing the babies, then a falling stork or declining stork population should lead to decreased number of babies in West Germany. Well, and I found another graph actually, which is measuring the amount of DDT in breast milk, and this is Sweden, this is not Germany, but this, and the red bars here are actually showing the same time period than the graph I showed before. So there's a falling amount of DDT in breast milk in the same time period where the slope kind of looks similar. So we could say maybe the decrease, this is actually, it's a very theoretical approach, right? This is not scientific at this point, but it would extend our AOP to falling DDT exposure would be the key event 1. Exposure is not a key event in the strong AOP sense, but just for showing the principle, leading to declining stork population, leading to less amount of babies. So what we have in here are 2 key event relationships that needs really proof, right? To see if this holds through. So this is really testing the hypothesis if this is a correlation or if there's causality behind it, right? So when you search for DDT and stork in PubMed, you actually find a publication that shows that summation operator DDT is 174 nanograms per mil in painted stork. This is a certain stork. So DDT can be found in storks, but if you continue, you find that DDT, as you know, is an endocrine disruptor, leads to eggshell thinning in birds, and actually leads to bird population declines, which then shows that for the key event relationship 1, there is really no literature support. So it's the opposite. So we would say that doesn't make really any sense. And doing some research also on what about storks and babies, then in the literature it shows that storks have really rarely been seen at sites of baby delivery, which, you know, I mean, the stork has to be present to bring the baby. So that makes us think that there is also no literature support for the key event relationship number 2, which actually says, shows that the lack of causality defeats the theory of storks with kind of an AOP concept. So this is something to bring the concept a little bit closer to the students. I like to use this example because, I mean, if you want to research about it in the web, this paper, this Nature paper really has been discussed a lot in epidemiology. Okay, so this was a little bit to make you smile in the morning. Now I'm coming to a little bit of academic research, and well, I'm a neurotoxicologist, and my main field is developmental neurotoxicity, and so we're having to deal with the brain. And the brain, when you think about the AOP concept, is a very, very difficult thing because there are many, many molecular targets in brain. And as we heard before, so an AOP needs one molecular initiating event going to one adverse outcome. So that's by definition, it has to be linear. I mean, think about all the different targets, and just modifying each target in one way would be one AOP, but if it leads to different adverse outcomes, it's multiple AOPs. So we're looking at such a high number of potential AOPs that you could build just by looking at the molecular assembly of the brain. So this is one thing that makes the AOP work difficult in brain. Yes, I mean, not impossible, but something to think about. And the other one is that during brain development, you're not looking at like an adult tissue where you have one state and you change the state from physiology to adversity, but you have different developmental stages. And actually, in the embryonic phase here, or the fetal phase, which is a little bit later, or postnatally, where the brain continues to develop, you have really different signaling going on because you have the brain cells are performing different tasks. So you also have to keep in mind that depending on the state of development, your AOP can be completely different. An initiating event that changes something during the embryonic phase might not do anything after birth, or vice versa. So that makes it really complex, and the complexity was actually brought to my mind when I had to do my very first AOP exercise, and this was actually initiated by Anna Price from the JRC at ECVAM in Europe, and she said, well, we should really try and do the first effort on AOPs and neurotoxicity in an academic context. And so I helped her organizing this, and we got together a bunch of well-reputed scientists with different areas of expertise. And the task was, okay, we want to create 10 putative AOPs, and we want the experts to come to the workshop being prepared and bringing all the knowledge they have about these putative AOPs to begin with. And these were actually times when the AOP wiki was in the very beginning, and also the OECD handbook wasn't really there, and the ideas on how to build an AOP was really rudimentary. There were no key event relationships, for example. So it started out with you need to have the key events and show causality, yes, but it was actually in the very beginning, and over the last 2 to 3 years, the field has completely made a huge progress and defined the way on how to make an AOP. It's much, much more defined. At these times, it wasn't that defined. It was more like, well, you try to show the causality, and it was the first ideas. And it was so much work to prepare for this workshop that I thought, what have I gotten into? And we met, and everybody was feeling the same. They're like, this was so much work to bring all this information to the workshop that it was crazy. But everybody did their jobs, and in the end, Maurice Whelan said, well, this was a workshop that got so much output, I've never seen this before. Yes, because we all worked beforehand, right? And it was really interesting to go through this procedure to be able to now understand how much work is it really. So these 10 putative AOPs came up, and it's a well-cited paper because it was really the first effort, but now things have moved on, and this is why the documents become much, much longer. The AOP descriptions become much, much more detailed and much more focused on the advices that the AOP, that the OECD handbook really gives you for developing. So if you develop an AOP, you really sit there with a handbook. You sit there and you follow each point, and this is the way to do it these days. So, but besides that, then after this, really, I started to think, well, we should use this also for implementing it in our everyday research. And I'll show you what we work with. We actually work with developing brain cells that come out of here, the fetal period, that can proliferate, migrate, and differentiate. And I'll give you this short introduction so you can understand that this putative AOP that we actually assumed based on our lab work. So these cells work in 3D, they grow in 3D spheres, they proliferate in 3D, but then they migrate on the surface and they differentiate in different brain cell types. And to give you a little live view how this looks like, these cells, they don't migrate out randomly, but there's a lot of communication between the different cell types. So my feeling is when you work in vitro, you should work with the best methods available. You should have the best that resemble physiology, the most closest to the human than what exists. And my experience is that tumor cell lines, for example, they behave a lot differently than normal cells, and so you have to keep this in your mind. And also the state of differentiation of the cells is really important, because depending on the time period during development, effects can be very, very different. And so I show you this, I don't know if you can see it, but it's a nice staining where you see that these cells that migrate out, a lot of them resemble radial glia morphology. That's important for the AOP that I'm going to show you. So, and how did we come up with this? This was really a coincidental finding, which quite often leads you to new paths that are exciting. And what you see here is the effect of a green tea epicatechin. I don't know if you know this. This is epigallocatechin gallate. It's a compound that you find also put into Coca-Cola and chocolate, and you can buy at high amounts as food supplements over the counter. You can order it through the internet, and what you ingest there is in the gram amount. So there is, if you take food supplements, there's very high exposure. If you drink a cup of green tea, the exposure is much, much, much lower. But we actually used this to begin with because this compound was also described to inhibit the dioxin receptor and be protective against dioxin receptor mediated adverse effects. But what we saw when we treated our normally quite pretty neurospheres is that you see something, that you see these little arbors here, and also in the rat, so we can do a species comparison with this model also. It really doesn't look pretty when you compare it to what it should look like, so a nice even surface. And we were very surprised. We're like, we have never seen this before, what is this? So that was the starting point. And we saw that it decreases the density of migrating cells, it makes these big gaps, and it looks really, really ugly. So this was the starting point actually for our AOP, where you have the MIE here and the adverse outcome in the end. And I'll guide you through this, how this project develops by using the AOP. So we have disturbed neuroprogenitor cell migration and a lower density of migrating cells. This was our initial observation. And how you would call this in AOP development would be inside out. So you have a cellular effect that you see, and you develop it then by finding out, so what might be the initiating event and where is the adverse outcome, right? This is just one way of doing it. You can also start from, for example, from like the big toxcast screening programs on MIEs. You start from the MIE and then you work your way back to the adverse outcome, or you observe an adverse outcome and work your way to the front. It really doesn't matter what you do, but it's all about, when I disturb this pathway in this cell, what is happening, and is there anything happening in the organism at the end? Okay, so we saw this disturbed migration, and we came across a publication about affinity chromatography that showed clearly that this EGCG compound can bind to laminin. And laminin is one of the most prominent extracellular matrix proteins in the brain. So during development, you need attachment of the cells to laminin so that they can migrate. It's really important. So we thought, okay, if the compound can bind to the laminin, maybe then the cells cannot find the laminin anymore because it's masked. And that this could be one reason why we see this curious migration pattern. And what we did to find out if this is really true, we did, so this is exposing the neurospheres in vitro in the culture very normally. Then here we pre-exposed just the neurospheres and then plated them on the laminin matrix. And in the third approach, we just pre-exposed the laminin, but we did not treat the cells, and then placed the spheres on the laminin-coated surface that had seen EGCG. And as you see easily, when you treat the cells, nothing happens. If you just treat the laminin, the cells can't migrate anymore. So this made us think, okay, we're on the right path. It seems to be a laminin problem. And when you titrate laminin in increasing amounts here to the EGCG treated cells, here in the highest amount, you see that the green tea doesn't have any effect anymore. So titrating the laminin, increasing the amount of laminin, helps the cells to find something where they can migrate on. So that added to our AOP scheme that chemical binding to laminin by EGCG seems to be the molecular initiating event. So we were it took us actually more than a year to find this out. It looks kind of short, but it was a lot of searching. So the question is, if this is true, how does the chemical do it? So EGCG is a flavonoid which has a pyrrogallol and a galloyl group. These are these two groups here. So we used structural analogs of this compound that have less of these groups. So the epicatechin gallate, it's also in green tea, has only a galloyl group. The epigallocatechin lacks the gallate group and only has the pyrrogallol group, and the epicatechin has none of those. And when we used all these different compounds here with a different amount of groups, you see that the effect is the biggest when you have 2 groups, and if you have only 1 group, there's still an effect, but it's less, and the guy here without the group actually has no significant effect. So this made us think that through these Gallowil and groups, the EGCG can bind to the laminin and stop adhesion. And if if the cells don't adhere, you should maybe see something while you plate them. And this is why we did a time-lapse video. So what does the neurosphere do when you plate it on an EGCG-covered laminin? So you see that the motility is okay, the cells still migrate, but because they can't really attach, you can really see they can't get a grip, they decide to move actually backwards. So we have, this was pretty crazy when we saw this, because it really showed that, yeah, it's trying, the cells are motile, it's not something that the cell is dead, but it's when I can't grab and hold on anything, I just go back to where it's cozy inside. So supporting our hypothesis. So when you look at the ATI zone then, up here you see the laminin ECM, and these are the different integrins that actually bind the laminin for adhesion. And you always have a beta-1 integrin or a beta-4 involved. So integrins are dimers, there are always 2 subunits involved. And we checked, well, we should see an effect also on the integrins if there is a problem with adhesion. First we checked, okay, integrins are there, beta-1 and beta-4 by PCR. And then we treated the spheres with a beta-1 integrin blocking antibody. So it's a functional antibody that blocks the integrin site. So we thought if integrin laminin binding is involved, then when we treat them with the antibody, it should look the same than blocking the laminin with the EGCG. And this is what you see here. These are actually controls with compounds that act through a different effect on different pathways on migration. You see there are not these gaps in there. It's just shorter the migration distance, but the beta integrin antibody really makes it makes these strange gaps here, because it's really adhesion. So I think this is the first putative AOP that had something to do with adhesion. And also by using the beta-1 integrin antibody comparison to the EGCG, you also have less number of cells in the corona, and you have these gaps in the corona that we can quantify. So adding to our AOP, we think that the EGCG binding to laminin and the disturbed migration has something to do with integrin signaling, with integrin adhesion to the laminin. And then we went into the literature, because we only work in vitro. So you clearly need some also in vivo information. And actually there is a group that works with knockout mice, making beta-1 integrin conditional knockout mice that lack the beta-1 integrin in brain, and they also saw a lower density of cells that have been dividing during development in cortical layers. And actually what else they saw is a disturbed glial cell alignment in the brain cortex, and these animals do have behavioral deficits. So because we thought, well, our neurosphere, I showed you the radial glia image earlier, is actually perfect for looking at glial cell alignment. So why don't we look at that? And this is what we did next. I'm sorry that the images are so poor, so maybe it's because of the light, but you see that the radial glia, normally it's kind of straight, and when you increase the amounts of EGCG, you see that the cells start to become chaotic. So they're not aligned as pretty anymore. And well, thanks, but I... Are
you done? Well, wait, hold on 1 second. There.
Yeah, here you see it better. So they're nice and straight when you stain them. The green ones are the young neurons, and these are the red guys are the radial glia cells. And with increasing amount, you see that the alignment gets lost, that they become more chaotic. And this is actually what this group saw in the animals in vivo, but also when they took the cells from the mice brain into the dish, they saw something similar. And you can also actually quantify this by measuring the angles here in the neurosphere. And so this is a polar plot of the angle. So the blue is the control. Most of the cells are between 30 degrees in angle here. But when you go up with the concentration, you see yellow and red arising. So you see more cells are actually off the normal angle. And then you can also look at what kind of cells do we have here, oligodendrocytes, neurons, and these radioglia cells, which are also positive for nestin. And you see that with increasing concentrations, these nestin positive cells get fewer to begin with because they cannot adhere. But secondarily also the neuron population goes down while the oligodendrocytes stay pretty normal. So we found in vitro that also the EGCG leads to a chaotic GFAP cell orientation. If this will result in a lower, or would result in a lower density of BRDU cells in cortical layers in vivo, of course we don't know. We just have the idea that what you see in the animals with the knockout is to somewhat similar what we see in vitro. And yeah, so this putative AOP was actually published in this is a common article here in Neurotoxicology, where from a session where we presented this at the INA meeting 2 years ago. And but there is an original article actually showing all the EGCG work. Well, what about exposure? I think exposure is really, really important because it's not included in the AOP, but exposure considerations, of course, are very important because, yeah, toxicity it's made by the dose. And if you want to come from a putative qualitative to a quantitative AOP, at some point you have to include doses and thresholds. And that's probably the most difficult obstacle that we have at the moment, even if we have a lot of information, how to deal with all the information on different concentrations in vitro, doses in vivo, in the different systems, to find thresholds and turning points and tipping points. When does one key event really trigger the next one? How much do I really need of this? So that is very, very difficult. I mean, even here we know, okay, when we block adhesion, we get chaotic GFAP cells, but how much do we need of this to really make in human brain development to really have an adverse outcome and effect? This is really not known, but we did some kinetic, pharmacokinetic, yeah, we tried to do it this is not by computer modeling, this is just by going into the literature and finding out what we see in vitro. Might there be really a reason for caution in the in vivo? Am I too long? Well,
No,
I'm almost done.
Yeah.
Oh, okay, so I'm almost done. Okay, so how do we approach the kinetics issue? So if you buy these EGCG oral supplements, then you find a concentration in there of 0.1 gram per mil. And you are supposed to take 2 tablespoons, which would be a dose of 3 grams that you take per day. So from pharmacokinetics in humans, actually, there were experiments where they made humans take between 1.6 and 2 grams EGCG a day. They found a maximal plasma concentration of almost 10 micromolar in blood. So that's pretty high when you take these supplements. Considering pregnant women, which have a different body volume and blood volume and so on, we calculated that a dose of 3 grams, which is 2 of these tablespoons, would end up in maternal plasma between 11 and 22 micromolar. And from rats, it's known that when the maternal plasma concentration is 0.6 micromolar, you have fetal brain concentrations of 0.07 micromolar. So in rats, it's known. So assuming that the kinetics across the placenta is similar in rats and humans, and this is something that we don't know, then a dose of the 3 gram would, and that causes maternal plasma levels of 11 to 22, would end up in fetal brain with 1 to 3 micromolar. And like I show you, the LOX in the human neuroprogenitor cells in vitro for migration, adhesion, and this GFAP process orientation is between 1 and 5 micromolar. So we are very close to exposure levels. And I think this is not proving anything, but it's showing concern that with these food supplements that are really not tested like chemicals, you should be really cautious during pregnancy. I mean, this is something that we concluded actually in the publication. So, and here I have summed it up just really briefly. So the high concentration, this is not drinking green tea, it's really the high concentration food supplements disturbs the adhesion and migration of neuroprogenitor cells, and it seems like binding to the laminin, which are these gray crosses here, that prohibits the binding of the integrins to the laminin in the radioglia cells. This is causing a disturbed adhesion and an altered radioglia alignment. And I showed you that in vivo, I told you that in vivo, the altered integrin function causes structural and functional neurodevelopmental defects in mice. And the pharmacokinetic considerations call for caution of high-dose food supplement intake during pregnancy. So this is kind of how we're trying to use the AOP concept really also in the academic everyday lab research. And what I tell my students, well, you know, it's a different way of thinking, but you always have 2 papers out of your primary lab work. So this is something, you know, I mean, every student wants papers and every postdoc, and so this is something you can actually write your primary paper, but then put it into the AOP frame, put it into a context, not to the OECD level. This is not doable, I would say, but to try to make an hypothetical or putative AOP out of your primary lab work and find out where are the gaps. You know, this is something that an academic lab, I think, can do if you put effort into it. And I would like to thank, especially here, Dr. Marta Barench. She's now got an academic position at the University of Barcelona, so she's very happy because she is from Spain. She spent quite a few years in my lab and worked on this. And I would like to thank you for your attention, and if you have any questions.